Can you hear me?
I can hear you. I can't see you though.
I'm laying on my couch. I'm like, crap.
Oh, wait, here, there, I see you.
Mm. Is that okay? Is this view okay?
Yeah, I'm gonna fix everything. Let me see if we're. Yeah, we are live. Hi there, Kevin. Nice to meet you, Lindsay.
I'm here.
Okay, I just have to tweak a couple things. I don't have a producer here. I'm just
We're
doing the best I can.
winging it.
Here, hold on. Let me. All right, that works. So we are here today with Lindsay, a nurse who was injured by the Pfizer shot. Is that right, Lindsay?
Yeah, actually today makes year 4. I took the Pfizer and here I am
Four.
today.
Oh my gosh, your
Yeah.
4 year anniversary.
Yeah. And then on the 23rd of this month was my first vaccine was 2020. So I was one of the first to get it. It was December 23rd of 2020. So that's when I first started making Spike and I'm still making it today. So 5 years of Spike and 4 years of being injured.
Have you made any progress or has it just been complete hell from the start? I mean, tell us sort of
So
what's happened.
the first two were Moderna. And this is where my case gets tricky because Pfizer is what injured me. The first two Moderna, I had like your body aches, fever, hot flashes. I had like a pre-fainting episode, pre-syncope. My second shot of Moderna, I actually passed out. I had all the same things. Well, we get to August, and I had transferred from the ICU and the ER, and we went to the cancer center. And a group of us went to go get our spike antigen levels checked just for fun because we didn't know what our spike levels were. We didn't know if it was protecting us or not. So we get to the clinic and we all get our things back. This is August of 2021. And my spike levels are at 2000 and the rest are like 300. I think one was like 20. It was like really low, and mine was way up there. So 4 months after that's when I got my Pfizer booster. And the reason I got it because work, they haven't mandated the booster yet, like the first two, but they were starting to push it and they were incentivizing for people to get them. We wouldn't be able to have to wear a mask in front of our patients, except for when we were in front of our patients. We got 40 hours paid time off or vacation. We could collect it in either a check for 40 hours or we could take paid time off vacation 40 hours. So they're pushing it hard. And at the time in my life, my husband and I, we were my ex-husband now, but we were going to try for a family because I haven't started yet. And I had just turned 36 years old 2 weeks before I got my first in 2020. Now I'm 40. So um, I got it because I didn't want to get it while I was pregnant because I just didn't want to put the baby at any risk. So I went ahead and got it, and then here we are today, still
Was
fighting.
Yeah, was the reaction pretty immediate?
It was the same as the previous two, but it never went fully away. I was able to get up and go to work the next few days. It took about 48 hours for it to simmer down a little bit, but it never went away. And then on day 10, it hit me like a freight train. And that's when I didn't know it, but I was in full cytokine storm. And
Mm.
my cytokines are still outrageous today. So hopefully that's where Kev comes into play and we can get my system. Right now, it's a battle between my nervous system and my immune system
Mhm.
because my T cells are exhausted. My cytokines, my interleukin 6, which is your main pro-inflammatory marker that we've been looking at across the board with us, it was 48.8 and normal is 1 to 3. So
Mm.
that just cascade. I think I had 11 out of 14 activated at the time. I
Wow.
haven't checked them yet, but I'll check them before I go to Japan for sure, just to get another baseline. But we have 4 years of panels. We
Mhm.
have 4 years of videos. We have 4 years of labs. Like they cannot dispute my cells. And that's why they hate whenever our faces come about because it's something that they have to face the music now because there's too many of us. And we want to know why the heck are we still producing spike protein? And we need to get it out of our bodies because it's killing us. We're dying. I'm dying every day. My quality of life sucks.
What have you tried thus far?
Oh gosh. I've tried a lot of stuff. Dr. Corey was is still my provider. He's one of my many and Sabine as well. But some of the things I haven't tried, let's put it that way. I haven't tried like the high cost vitamin IV infusions because I'm pretty good about getting my supplements through food first and then what I'm not missing get afterwards. And that's the thing too. I have all the labs, so I know what my genes look like. I've had the RNA DNA integration tested. I know what my gut's looking like, and I know what's going on. So those three main panels with your micronutrient, I kind of can attune my supplements to what I need. But I haven't tried Evo. That's where it's kind of similar to what we're doing, except it's not on a cellular level at all. It's more of a basic level to get regular gunk out. Maybe if you were having mild issues, it would help.
Mhm.
I haven't tried that, and I haven't tried hyperbaric oxygen chamber just due to the clots and the risk of me possibly throwing a stroke or having a heart attack from it. So those are my two main risks right now.
Okay. Well,
Yeah.
Kevin McCairn is here with us. He's a neuroscientist. He's in Japan. Are you there, Kevin? I think I
I
see
am
I
here.
see
I
you in
don't
the
know if you
corner.
There he
can.
is. There
Yes,
you go.
Yeah.
I've been listening in the whole
For some
time.
reason, you're in this tiny little corner on the screen.
Yeah,
Oh,
that's probably because I was streaming at my end, and if I do that, you can probably see me better now, right?
Yeah.
But then other people will just have to listen to you on my stream if they want to watch. You two are far better looking than me, so you should all go over and watch Mary's stream instead of this one. But the yes, I am a neuroscientist. I was ensconced in academia up till COVID. COVID happened, and then I, like many others, had my world turned upside down because of it. And I came out early, saying this wasn't natural. It was the process of or the output from many years of dedicated work in the biowarfare space, and that gene transfecting the population en masse with viral spike proteins, and my particular bailiwick, which is amyloids and amyloidogenic attack surfaces, was a incredibly bad idea, and you're going to end up with people like Lindsay as a consequence. And perhaps I can. I've spent a lot of time looking into sort of like the pathophysiology of COVID, and although I think that's been helpful to people, it's only till recently that we've been able to approach something approximating a reproducible therapy that has metrics associated with it that I can point to and say, this is the difference pre and post treatment. And with respect to some of what Lindsay was saying, I've been testing people's blood now for amyloids for a year now. It's over 100 patients, 400 samples. Many of them have done all the recommended therapies, be it ivermectin, hydroxychloroquine, um, NAC, turbo NAC, you name it, even EBU,
Mhm.
in fact, doesn't work. In fact, we have a family of four right now here in Japan who literally went four times for EBU on my recommendation. One, because their son, well, it's kind of a complex, well, it's not complex, but they had seen me talk on Steve Kirsch VSRF, and the mother sent all her children and themselves samples to check, and they'd been all been vaccinated by the same Pfizer dose. And I checked the son, late teenager, 17 years old, and was loaded with amyloidogenic signals in his blood. And we know that teenage boys seem to be at high risk for cardiac problems post-vaccine. And so I made a point of calling the mother and saying, you should be aware of this. And then sort of we started down the path towards trying to do something. And it was in that time period that we began to get the first data back from EBU failing and moving towards dual filtration plasma apheresis, or DFPP.
Wait, say that again. Wait, say that slowly for everybody.
So dual filtration plasma apheresis.
Okay.
And so this differs from all the other apheresis techniques out there in that there's no plasma or donor plasma involved. It's a closed circuit, and the device comes tuned dependent upon the illness you're trying to treat. And it is covered in Japan for insurance for a few conditions, primarily autoimmune conditions like Guillain-Barré or pemphigoid, things like that. Real sort of life-changing illnesses, but it's not available. It's not covered normally. But we were fortunate enough to have someone sort of step in and sort of act as a guinea pig, or test monkey, I should say, because that's what I would use
We're all
previously.
guinea pigs, Kev.
Yeah, all test monkeys now. And what we found again was EBU didn't work, and dual filtration plasma apheresis did. And in parallel with that, what I discovered in my lab is that we could take stem cell growth factors, and these are growth factors derived from, so when a child drops its tooth as it matures, there's a sort of remnant pulp cavity in that tooth, and they can recover stem cells from that pulp cavity that's left, and then they can culture that out and then harvest the signaling factors from that cell line, and then they can infuse that, usually intravenously, as a therapy. And what I'd found, I'm happy to sort of show the data if you like. It's probably easier to visualize rather than me just talking about it. But in the presence of spike protein and a seeding clot, the stem cell growth factors were able to inhibit completely the formation of new amyloidogenic clots in vitro bench testing. And it's kind of a adapted, so it's called real-time quaking induced conversion, and it's used to look for prions. Basically, that's what the test is derived from. And so I actually had that bit of information for quite some time. And so we now had this ability to stop the progression of the clots, but it's only until the latter half of this year that we've had the proof that the dual filtration method works. And in doing the dual filtration method, there's the filter column that you use specifically for the treatment condition. And in this one, we're using I picked it basically, but one for polyneuropathy and sort of autoimmune attack against the nervous system. And so right now we had 2 very successful patients, and right now, fingers crossed, we're in the middle of the next 4, and 7 will be coming in a week, 2 weeks, less than 2 weeks from now, actually. But
just real quick, is it just to try to visualize what's happening? Is it similar to dialysis where you're just filtering the entire blood system? And
yes, yeah.
So time-wise similar to that? Is that like
yeah? So this is
hours and
you have to sit there for a few hours, but I probably should share my screen and just go through
the vampire procedure, the big catheter.
Yeah.
The catheters are bigger than my neck, Mary, and they're going to go in my jugular. So that terrifies me in itself, because I'm worried about, am I hemodynamically stable enough to withstand the fluid shifts, because it's going from intracellular to extracellular, so forth. But basically, it's separating the plasma and the blood, and it's catching, being selective of what it catches, such as cytokines. And go ahead, Kev. I'll let you go ahead. Sure.
Yeah. Just give me a second. I'm trying to. It did say share. All right. So now I want to be able to. I don't have it open. Surely
Does
I
it
do.
have to be done multiple times?
So currently we are doing it twice. So we've worked out a sort of 14-day treatment protocol, intensive protocol, and we're still early days in this, but the consensus at the moment is to, as the patient arrives, to try to do the filtration as early as possible. Then every day give stem cells, and then at the end of the 2 weeks, do another filtration, just to make sure. And I'm sure I had that thing open. Where do I
The family
see?
that's there now, they're actually watching, and I just wanted to give them some words of encouragement and hope. And thank you for doing this because, like I said before in many of my posts, if you don't know me, this is bigger than ourselves. This is, the entire population was poisoned, so we have to figure out how to help us. And thank you guys for stepping up and getting it done.
Yeah, so hopefully I got a share there and you can see that real quick. If you can confirm. Should see
Yeah.
just like a bar graph or whisker and
Yes, we can
box and
see it.
whisker plot. So that's the chart that I was on about before, where it's basically just a measure of intensity in these little experimental wells. And we're just looking to see, and the therapeutic result, which is the three bars to the right, are just different volumes of stem cell growth factor that I've used, and all were effective. But if you can see that there is a dose response with the increasing amount of stem cell growth factors that we do use. And just to give you some idea of how we do the apheresis, so you can get it's not um, it's not available in the US. It's not an FDA approved treatment, but it's quite common to see it in Asia. And it's used in regenerative medicine often.
Mhm.
And the idea is
Why is it not available in the US?
you'd have to ask the FDA that, and it's a non-pharmaceutical treatment, right? So um, that's probably you'll find a lot of the answers there.
Are
And
there companies, are there people that have tried to get it passed and have been shut down?
So here's it's possible that Heather, who was going to join us today, but unfortunately wasn't able to. There may be one clinic in the US who's doing it, but whether they're doing the same approach as we are, I couldn't say. But the critical factor is the combination of therapies all at once. So which well, depending on the patient is going to vary, but I think in most sort of long COVID and vaccine injured cases that the polyneuropathy treatment columns are going to be the direction to go. I know in the case of Heather and Cody, he has a triple antiphospholipid condition, but in Asia, they specifically treat antiphospholipid disease with dual filtration apheresis.
Mm.
So there are columns specifically designed for that specific condition. But what you can see on the screen there is the triple catheter that's inserted into the jugular vein.
Mhm.
And why go into the jugular vein and not the brachial veins as you would in a normal regenerative DFPP approach? And just we want to be able to clear as much from cerebral circulation as possible and as quickly as possible. And so you try to hit as large a vessel as possible that's directly linked to the brain in this instance. And if you try to do it through brachial limbs, you're bulk filtering, trying to do the whole body instead of getting this targeted filtering done as the waste is being drawn directly from the brain in this instance. And on the panel to the left, I mean, this is data that we just got in the last week. On the left-hand side of that image, you can see it. So I'm holding it up to the light. You can see the plasma pre and the plasma post filtration, and you can see how the pre is way more cloudy, and the you can practically see through the plasma after the filtration procedure. And so this is sort of, well, prima facie evidence of the ability to clean and scrub the blood like the protocol says that it will. And again, the critical factor here is, and this is something that I haven't spoke about because I only got that data recently as well, was you're not taking donor plasma. And for the moment, I'm of the opinion that you should consider all blood products tainted until proven otherwise with these fibrin amyloids that are in the circulation that are a consequence of both the disease itself and what the VAX injured show. And so if I sort of move on. So yeah, any questions before I sort of show you sort of more
No,
granular
Kev, I
details?
wanted to piggyback what you just said about the plasma because we're seeing that with the injured and the
Mm,
IVIG patients. The
mm,
plasma donors, they're getting spiked because they weren't, filtering their bags. So a lot
mm,
of them have transferred over to subcutaneous immunoglobulins instead.
mm. Yeah, and again, I literally got that data just the week before Christmas now, and I was sent donor blood. So shout out to Safe Blood USA. They're a charity that's sprung up in Texas, and they're I don't know how much I want to give away without their sort of direct say so, but let's just say they're medical professionals that are directly involved in surgeries where blood transfusion is a large part, and they were concerned about donor blood, and I can tell you that I can confirm that it was relatively easy to find amyloid signature in the blood, in
Well, yeah,
donor
I want to
blood.
ask you about that. This testing for amyloid in the blood, how hard is that? Is that something available in the United States?
Um, I mean, you can. It's just that there are newer tests coming online right now, which use flow cytometry, but they're trying to sort of. It's the same game of try to patent something and then charge exorbitant prices for it, and but it's a relatively easy test if you just use old school pathology methods of just microscope the dyes and a bit of patience to sort of sit and look through properly assess a microscope slide. And those are becoming rarer and rarer skills these days,
I have a
unfortunately.
microscope in my office and I've actually, so I can just take
So
some
you need a
blood and
UV,
get
you
the
need a
proper
UV microscope,
a
so
UV.
ultraviolet, yeah.
Okay.
And that sort of just it puts it out the range of sort of your keen enthusiast in terms of budget, but the principles itself are very basic. It's Fireflavin T dye. If you want to get really fancy, there's one called Ami Tracker. But I'm, the condition, like SARS itself and COVID, was identified as a disseminated intravascular coagulopathy very early on in its emergence. The fight has been one to recognize the claims made by people like Lindsay that, hey, You essentially have just exposed us to an unknown amount of spike protein that in some cases seems to be in permanent production. And two, well, there's just this back and forth all the time between the more lurid stuff like nanobots in the vaccines and graphene and that sort of thing to viruses aren't real. And that's often that for the first 5 years, I've literally spent hundreds of hours um, battling that type of I don't know what to like psychological operation, I guess I would say, because people who have hurt Lindsay like that sort of narrative to take a hold in social media because then it obfuscates what's happened to Lindsay, right? And then Lindsay gets sort of lost in the noise of all these clamoring voices. And even this week, I can't even remember. It was Jicky, and I'm sure you know. I don't have to say who he is, but there was another thread about people talking about graphene being in these vials, and I know this. I'm one of only two laboratories that I would trust. There was myself, and there was the working group, German working group, they called themselves, who did the required testing, which means Raman spectroscopy, scanning electron microscopy, and they went even further and did nuclear magnetic resonance imaging and mass spec to show that there's in all the vials that we received, and I've got a freezer full of vials, there was no vaccine. What the primary problem is the one discovered by Kevin McCairn, which was the DNA contamination. And just from our lab, shout out to Charles Rixey, a vial from that was in my freezer just got the hottest signature ever for DNA contamination in it. And you've watched this as the regulators and the pro-vax crowd initially tried to dismiss the claims that there wasn't vaccine, sorry, there wasn't DNA in the vaccines, and then they've step by step had to walk themselves backwards and admit that yes, there is DNA contamination, but now even if it exceeds the regulatory limits, there's no reason to worry. And the SV40 nuclear localization signal that's been identified is nothing to worry about, and that doesn't in any way explain why Lindsay's permanently producing spike protein right now. And so these are all facets and fronts of a very complex fight that, in Japan, I don't feel the American political aspect of it, right? So all that's happening with Congress and what have you. I know Charles Rixey's sort of once a Marine, always a Marine, and he's there fighting pitbull like for his country and his people, but he is involved in Congress and trying to get RFK into power, all these things. But my position is one of I think it would be incredibly unlikely that you will get the pharmaceutical industries or the regulatory agencies to in any way admit culpability at the moment because culpability is so large because of that scope from the biowarfare field to that just infecting billions of people in and of itself. And then the hundreds of millions that have been exposed to genetic engineering tools as a public health measure. There was no scientific or medical rationale that justified that move, yet they did it. And so of course they'll just keep kicking the can down the road. Trump's not going to be in office forever, and neither is RFK. And so what's left to do is to it's probably have some degree and sense of realism and just think probably what we can do is get effective treatments to the forefront. And that's where I think I'm hoping we've finally sort of cracked that particular nut in this instance, but it's been a long, hard slog. And if we can make a difference with Lindsay, then I'm sure we can pretty much repair most people, get some functionality back to everyone.
How many people have you treated so far?
So we've had 2 done. There's 4 in process right now. They're watching right now. And we've got 1 coming in on the 12th, and hopefully more. And then we've got Lindsay, hopefully. We'll look after Lindsay. But Lindsay doesn't have to worry about all the living expenses and stuff when she's here. She's essentially my guest. What we have to do is to raise the treatment still costs money, and that's a unfortunate fact until we can maybe get I don't know, if the fund that's available for vaccine injured could be somewhat pointed at this therapy in some way, that would go a long way, I think, to actually, one, having the therapy disseminate to hospitals that could do it, and 2, just helping who can put a number on it with respect to injuries, whether acute or the long-term sequelae of exposure.
Are there risks to this procedure?
Well, obviously, putting a catheter into the jugular is always has some risk, but it's well tolerated. Second patient was someone who was really, and she was a severe long COVID patient who had um, was right on. She was developing symptoms of Parkinson's. She had major depression. She was really going downhill very fast. And within hours of her doing the treatment, she was like, "Oh, the brain fog," which was one of her primary complaints, was just that she was, um, she just couldn't formulate thoughts the way that she knew she could. And she said that lifted within hours of the first treatment.
Mm.
And unbeknownst to me, I want to say it was last week, the Twitter space that they do stop the shots. She was there on sort of like the last 20 minutes. Tamara, her name is. People can go and listen to her testimony of the treatment in Japan, and just how it changed her her symptoms. Now, you asked about complications. So just with her, the second approach didn't do jugular. They went femoral, and they nicked an artery, and so she had an overnight stay just as they had that compressed to make sure that it didn't cause any further problems. But short of that, her symptoms have been holding. Our first patient, I have his slide on my desk, waiting to test for this month, with respect to amyloid burden. But from his first treatment to now, which is approaching 4 months, I'm going to say he's amyloid free, normalized cytokine panels, and the every metric that we have so far is that it's worked. And in the really acute case of long COVID, which it does exist, and people are in a bad way if they do get it, and the teenager who essentially was amyloid loaded post the vaccine. I'm happy to say that, yeah, so far it's all working great, and this isn't it's not a type of, I don't know if they're like, don't get me wrong, the nutrition part is important and the supplements, etc. But that's all part of having a sort of clean lifestyle, and Same goes for sleep, everything. All those factors are important for sort of maintaining your health. But there's become a sort of blind faith and almost alchemy type approach to what you should and shouldn't be applying to try to remove these amyloids. And I can tell you, I've I have many samples from people who are very, very ill who have gone through all these protocols, color protocol, etc., etc. Does not remove those clots.
Well, yeah. What about nattokinase? Does it
Doesn't
dissolve?
work.
You
No,
don't feel like it works.
no, no. I got too many, and it doesn't. It hasn't worked in the lab when I've tested it against lab grade nattokinase. And maybe I in this instance I was testing it against um the embalmer clots
Right, right.
that Richard Hirschman had. So maybe those clots are just too far gone to be able to do anything,
So
and
that
maybe nattokinase helps in some clotting conditions, but those amyloidogenic ones,
so the cadaver clots, those are amyloid. Has that been
yeah, oh yeah, yeah, I can,
analyzed and
yeah,
determined?
I've done that.
Yeah.
Mm, mm.
Okay.
I can show you that data as well if you like. But to there's two principles from which I to just sort of summarize, and then I'll show in what it looks like in the data. So two principles which I'm sort of using as the metric and guide stone for treatment in this instance, which is one, the stem cell growth factors demonstrably on the bench stop that amyloidogenic cascade and the misfolding of proteins. And I'm as confident as you can get. Those are high p-values or low p-values, depending on which way you want to look at it. But it's not chance that it's doing it. And um, my problem is I don't have good a good um, a good answer as to why it does that, because the stem cell growth factors is it's not um, one peptide that's being secreted by those stem cells. It's like 100 different peptides, and what synergistic combination stops the stops the conversion? I don't know, and it would require institutional level funding. And you have to. What I'm doing is all crowdsourced and crowd-funded to get to this point. And so
Are you measuring spike protein?
I can do spike protein, have ELISA in the lab. I can do, so we've just got what's it called? I've forgotten the manufacturer now, but so I can now do, I've got ELISA, so I can measure spike protein. I can do cytokines, Luminex, Magpix, that's the one I was looking for. Quantification of cytokines. I have the like RT-quick type approaches, and we also have a citation 5, Agilent citation 5, which basically automates now the reading of the slides so that you can get so currently when people call me, if they've sent a sample in, the only test that you get back, when you asked previously about the test being done in the US, yes, it's done, but it's only a qualitative answer that you get back. So that's Dr. Vaughan in Alabama. I know who's doing it. And basically, he'll grade you between 1 and 4, as 1 being normal and 4 being severe. And um, but it's based off the judgment of the person looking down the scope, right? And so coming with that, you have the requirement of, well, one, you need to know what normal blood looks like, and then you have to build out a picture of how that maps to people with and symptom profiles. And so I've got quite attuned to that. But there's still some degree of how do you say? Yeah, it's I Go on.
Well, I was going to say, I mean, the only thing we have available basically to give us some sense of what's going on, in my opinion, is the spike protein antibody test. Does that correlate with spike proteins? And does that spike protein level correlate with amyloid? In other words, is a high spike protein antibody level, which is super easy, easily available. You don't even need a doctor to order it. Is that reflective of
It can be.
spike protein and amyloid?
It can be. But the problem is with amyloidogenic disorders that once initiated, the primary sparking agent, like spike protein, can be long gone, and the cascade will continue once it's left because it's a protein misfolding disorder. And the problem is that it's so in my field, like the only reason I sort of had any expertise as to approach this problem was that I worked on modeling Parkinson's in monkeys prior to COVID. And so Parkinson's is an amyloidogenic disorder of alpha-synuclein in dopaminergic neurons. And so the principles, and in the field of neuroscience around 2013, there was this sort of conceptual jump made that Alzheimer's, Parkinson's, all the sort of neurodegenerative disorders that we're all familiar with, they all share a common mechanism with Creutzfeldt-Jakob, the canonical prion-like disorder, and that is this protein misfolding, mistemplating phenomenon which occurs post-translation of the mRNA, and as the protein is supposedly in its mature form and supposedly doing its job. Now, when trying to make the analysis and look for misfolded proteins in neural tissue, you're basically taking a very thin slice of that tissue, you're staining it with this dye, and you're looking for subcellular inclusions, right? So you're having to go down to very high magnifications even to be able to catch the signal that you're looking for. And so the common one that most people would be familiar with is just Lewy bodies and where they aggregate, right? These are micron-sized inclusions in sort of a relatively large cell anatomy, particularly if they're large motor neurons, for example, or complex neurons like dopaminergic ones. And so the protein, the amount of protein that's involved is actually incredibly small relative to fibrin and fibrinogen, which is the primary culprit that we're seeing with this blood-borne misfolding disorder that Lindsay has right now. And you can see why that would be a juicy target for bioweaponeers to aim at, particularly if its short-term effects are non-lethal, right? So this is where you start to get into this area of why were they doing this research in the first place? And haven't we made biowarfare illegal in the 70s? And there was this gray zone of incapacitation agents that allowed this ongoing research to be carried out. And in that case, they could always keep sort of this circular money train going of, well, we need to find these incapacitation agents out, and then we need to find the countermeasures for it, which was essentially what the whole EcoHealth Alliance story, I guess, if you want to call it drama, however you want to, crime, I think I would call it. But EcoHealth Alliance was a tiny fraction of a multi-billion dollar research program that was spread across multiple US military military industrial complex. That's the word I'm looking for, because it's serious words that I'm looking for, and involves DARPA, DOD, DITRA, all the big universities, they were all pulling from these government money train, well, gravy, I should say, gravy pit, and the um, and the outcome was eventually, yeah, it was going to be either leaked or used, and maybe that's a topic for another day, but the if you're trying to think of an incapacitation agent, well, the one way you want to do that, if you just think of you want to choke your enemy somehow, right? And so if you can reduce his oxygen carrying capacity, and you can do that generally in a non-lethal manner, such that it doesn't raise too many suspicions, that becomes a primary focus, and why I think you're seeing or why we've seen the emergence of that spike protein the way that we did. And the fact that it had, this was recognized very early on, it had very discrete, novel amyloidogenic epitopes on it. And I spent a lot of time, because my concern was that those epitopes were, and again, this is, not all academia is bad, right? So I know it's easy to sort of brush it off, and I'm very jaded of it myself, but it's because of those areas of research which were able to identify those areas that we came to know that, particularly, I want to say it's amino acid sequence 561 to 581. I have to check my notes. It's been a few months since I've actually given that presentation, but I'll scroll through it right now, actually, just check. But there was a 20 amino acid sequence in there which was highly catalytic for conversion of the prion protein into its scrapey form. And in fact, they took all these different, so you could basically order 20 amino acid segments, and they took all the 20 amino acid segments of the spike protein, tested it, and there were some that were more amyloidogenic than others, and that it would sort of target the canonical proteins that we see that go wrong in Alzheimer's. But when they looked at conversion to the prion protein, it was orders of magnitude more reactive for conversion of prion protein to its scrapey form relative to all the others. And I can probably just quickly find that.
Hold on. What, the scrapey form?
Can you explain
what that is?
Yeah. So if you see on the screen here, right? So so a shout out must go to this lab, right? Per Hammerstrom and Sophie Nystrom in Sweden. And they were the first that came out. They had a preprint. This is how bad things were with respect to censorship, right? And I don't want to do a woe is me, right? I'm censored left, right, center. You can't click on my Twitter profile. You can't search my name on Twitter, that sort of thing. That's still all in place.
I had a very hard time finding you.
Yeah, and that's all still in place, right? And so people should keep that in mind. And like I say, just because things have changed a little bit now, doesn't mean they can just turn the dial back up for more censorship if they're still sort of holding me captive somewhat. But anyway, back to this. And so what you're looking at there is Hammerstrom's lab taking and looking at the lag time to convert the prion protein to the scrapey form. And what you can see, if I don't know if you see my mouse moving, right? Down here.
Yeah.
Do you see it? But anyway, there's a 20 amino acid long sequence on that spike protein, which is included in there's a direct amino acid. Well, in fact, you could say codon optimized, so it's not even direct amino acid, right? That they've made it so that it would be produced. That this 20 amino acid segment catalyzes the conversion of the prion protein into its scrapey form. And
so explain what a scrapey form is, though.
Misfolded proteins. This is how I do it, Kev. You say your body makes prions normal, And when they
Yes.
start to misfold, that's when you have the scrapey, the bad prion part, right?
Yep.
Okay.
Yep. And the best way to think about it is mad cow disease or chronic wasting disease in deer in the US. And the and so, for sort of 5 years, I was sort of, trying to bang the drum of they've just they've just released an agent that has this capacity baked into it. So in terms of, okay, you may get the immediate incapacitation effects, but there's a long-term sting in the tail, which is that there's likely to be this large increase in neurodegenerative disorders, particularly as they relate to things like Creutzfeldt-Jakob and the other, well, name your dementia of choice in this instance. But
I mean, and it's been a while since I studied this, but I thought that these amyloids, the prions, they deposit into the tissue and they kind of get stuck in there. And
mhm.
are they, I mean, is it really that easy? You can just filter the blood and they come sucking out, or is it
So let me just do this. I think this is I'm going to point people to this manuscript. So if I stop, or can I share a different screen? Yeah, I just want to point to this. And I think it's important for what the, because it relates to the therapy that we're talking about now. And it's this manuscript, which is fibrinogen mitigates prion-mediated platelet activation and neuronal cell toxicity. So it seems that the body has a natural defense mechanism in sort of sacrificing fibrin and fibrinogen as um, a sink, if you like, to stop the impact of misfolded prion proteins catalyzing their way through the body. And in a way, that kind of, in evolutionary terms, that kind of makes sense, because you would, as a I guess as a tribe, you want to limit catastrophic illness, something like a Creutzfeldt-Jakob, and if you can do that by um, utilizing a bulk protein that's there in some of the, one of your most common proteins is going to be fibrinogen, right? Because it's just circulating all through your body there to prevent clots, among other things. And if this article is to be believed, to mitigate against the spread of prion proteins as well. And so it's quite possible that because of the this mechanism, that um, uh, we're seeing this lineup, if you like, where we look at the blood and we see this misfolded fibrin as these sort of amyloid inclusions, that it's that prion protein catalyzing epitope which is potentially causing it. So it might be that there's some conversion to prion protein, but the fibrinogen, the body's internal fibrinogen, is buffering that cascade, if that makes sense.
well, can we give people fibrinogen, extra fibrinogen? And
Well, I think we can take out the misfolded fibrinogen, right? Which is what I can show you here. So, oh, so you asked about the hang on, I have to switch share to this. So you asked about the embalmer's clots, right? So I can just run through that quickly, right? So that's literally under dissection microscope. Then we cryostated it. This is it under normal light microscopy at 40 times magnification. Now, one of the defining features of the microclot phenomenon, it was known prior, and a lot of credit has to go to the laboratories of Pistorius and Robert Kell. And they were working on amyloidogenic microclots prior to COVID, and there was this sort of increasing degree of evidence that conditions like ME, CFS might be being driven by this microclotting phenomenon itself. And they had shown that the the microclots are autofluorescing, right? That's one of their key features that you can look for when you look at them under the microscope. And if you look at these embalmer clots, right? This is the edge of like the glass slide, and that's without putting any dyes on the sample. It's just hitting it with blue ultraviolet light, and it's fluorescing green, right? So it's auto-fluorescing with no dyes added. This is with dyes added on the left-hand side. You can see now that it sort of it's got this polka dot appearance, and all those little dots are likely those microclot aggregations, I would say. And they've sort of, in this instance, all this person was one of the unfortunate ones who's just all glued together. And why these people were not everyone else, again, we don't know. It's not, I've held these clots in my hand. Again, I've got a freezer full of them if anyone wants some. And just to sort of amplify the difference, sorry, you can just see how brightly it's fluorescing relative to, this is at 40 times magnification. And then I was able to do scanning electron microscopy, and we can go down in size, in scale, sorry. And this is looking at the fibrin at 5,000 times magnification. And what you can see, fibrin should have a smooth pasta or spaghetti-like appearance, right? And you can see in this that it's highly twisted and nodular, and that's sort of it's one of the sort of indicators that you would want to look for if you had a candidate for misfolded protein. It's just that because we have so much of it to look at in this instance, it's easy to go in and get, I say easy, it's not easy, but in terms of getting the picture like that, that was like, I don't know, the anatomy microscope gods were shining down on me on that. And just as we got that picture, this manuscript came out in Nature, which is Fibrin Drives Thromboinflammation and Neuropathology in COVID-19. And if you look at their work, you can see, so the left is normal fibrin, so it has that smooth pasta-like appearance. And then on the left, you can see that it's all nodular. This is after exposure to spike protein. And you can see that it's covered in all these nodules, like I show here. And so no heavy metals involved. I was able to test all that. What else?
Is there an imaging study where we could tag amyloid and do, like a PET scan, but for amyloid?
Yeah. So there is work being done on that. The only issue I have with that is that it's a radioactive tracer, and I sit in the pocket of one, they tend to come with any contrast agent for imaging, just comes with a whole slew of extra risks added to it. And I always tell people, do not take gadolinium if they offer you an MRI. It's not needed with the high power MRIs we have today. It was to be only in very specialized cases now that I think we need it. And if you're using PET ligands, those are, again, it's radioactive tracers. And that might be, again, it's cost benefit. And if you can get the answer from a blood draw and a microscope rather than exposing the patient to a radioactive isotope, then I think that would be the better option in this instance. But I want to say it was Princeton that was doing the radio labeled fibrin marking, but I may be wrong. But from the cadaver clots to someone like Lindsay to people who just had COVID, the primary distinction is one of a buildup of amyloidogenic fibrin in the blood. And the question is, can we get rid of it? And so here, looking on here, you can see the pre and post plasma. And um, what you're looking at here is someone's blood. So this is using whole blood as a measure, and in this instance, I just sort of putting a false color overlay to look for areas of intensity, and this is that autofluorescing phenomenon, and um, you can see. And though that autofluorescence is likely this so microclots and indicative of inflammatory conditions. And so, um, and this is post DFPP, so
Oh, wow.
radical reduction in signaling. This is microclots in that individual, and then this is post.
Wow.
And I don't know, I graphed it out the other day, but it's such a noisy graph that sort of difficult to make head nor tails of it. But we see reduction. It's probably easier, just like in the original format. So that subject or patient, as soon as I saw their blood and this degree of clotting, I said, you need to go and get a cytokine panel done. Okay, which they did, and now we can do that. But they came back with one, two, three, four, five, six, seven cytokines, all out of range, all high. And on his last one, which was November, um, some are showing lower than
Yeah.
normal range, and they're all normalizing.
And do they feel better?
Yeah, less chest pain. And if you think about what are the sort of what would be the first symptoms that you would expect from your blood being loaded with microclots? Things like brain fog, and also just, transient um, transient cardiac angina like pain, probably. And I know from the only reason I'm so confident in saying that is because I have to suffer it myself. I got COVID in February. It left me with two pulmonary embolisms, and now my blood looks like that.
Whoa. Did you get the
Yeah.
shots or
No,
just from
no,
COVID?
just from
Oh,
COVID.
wow.
Yeah, I did everything everyone says you're supposed to do when you get it, and yeah, it was in that pocket sort of a month later after sort of like the initial illness had passed. I'm sitting where I am right now, and yeah, I had to call the wife, take me to hospital, and they found, yeah, either side
Wow.
on, and yeah, you can look at my blood right now, and it's filled with microclots. I'm just trying to find time to get myself into this therapy as well. But
How much does the therapy cost?
so the hospital, after the first two patients, unfortunately, has raised their prices
Mm.
a little, for just because they were. I don't think they were prepared for the scope of work, and just they're having to deal with foreign patients in this instance. But it runs about $5,000 now per treatment.
Per treatment, and there are two
Mm,
treatments. Is that
yeah,
the
yeah.
okay?
And the stem cells, you can just budget $1,000 a day, $5,000 for the DFPP, and it's still orders of magnitude cheaper than anything in the US that you would get. Um,
Yeah, so the stem cells, is that how is that done? Is that every
so that's
day or
delivered IV every day,
okay?
and generally the sort of target dosage right now, they just basically charge per cc, and they're giving 10 cc's of stem cell growth factor. Um,
So that's 10. So it's basically $20,000. Is that
let's see. So 14 days at yeah, let's say 25. I don't know. Just you have to pay for a translating service whilst you're here. So to get the stem cell is just a quick in and out procedure. For the DFPP, you should just think the whole day, basically. It's less than a day, but the just budget a day. And but like I say, the effects are immediate and stand out. There's no waiting around. You don't have to take it for months.
Right.
It's pretty
Sounds
striking.
sounds like it's pretty low risk.
Like I say, there's always a risk with putting a catheter into a major vessel, but in someone like Lindsay's state, who just has no quality of life anymore and has been destroyed by these corporate avarice. But yeah, I wouldn't hesitate in saying she should be next in line. And I'm myself and Charles are trying to do everything to sort of facilitate. And thank you to everyone who's sort of contributing to her Give Send Go, so that she can get treatment. And maybe we don't have to um push and try and slam everything into 2 weeks, right? She's can be here as my guest, and she doesn't have to worry about a roof or meals in that instance. And so, if it takes a month, 2 months, 3 months, we can just take our time, and maybe if it's a case of raise enough so that she can do like the protocol that's working right now, and then work towards if we have to do follow-up treatments for someone as severe as Lindsay. But like I say, our second case was really, like I say, on the if she'd left it 6 months, a year longer, she would have been in a severe, like she would have, Paul Parkinson's would have probably taken her by that point. And I can just to show you so this is the most recent data. This is the young lad who was vax injured, and this is his. So in this case, I'm just looking at plasma, and this is his pre-DFPP amyloid signaling. And so you can see all these bright inclusions, but also the background signal is very bright as well. And then post DFPP, that's how it looks. And then now we're able to do quantification, and we can see that there's a significant drop in inclusions in his blood. And so as a it doesn't get like I say, I've always worked in sort of preclinical research, right? And it doesn't you don't get a clearer signal than that one. And I've seen them tee off on treatment paradigms and invest hundreds of millions of dollars on far more ambiguous looking signals.
Well,
So,
I have to admit, I was a skeptic, and but you've changed my
okay,
opinion.
that's good.
And there are about 6,000 people watching right now. If
Oh, okay.
everybody donated $5, we could get Lindsay to Japan, no problem. And I left
Yeah.
the Give, Send, Go in the comments of the live stream.
Yeah, and so if people want to get their blood sample tested, if you can give them this web address and send it, and then I can do a quantification of the stem cells, not stem cells, the amyloid inclusions in there. And in this instance, you just have to do a finger prick test and drop blood onto a slide, and that's enough for me to tell you if you have a signal or not.
Oh, that's very cool. How do people do that?
So you go to synapticlabs.com protocol on sending blood samples, and I'm just trying to find if I stop sharing, no, it doesn't give me the chat thing here. Yeah, chat. If I put this in the chat, do you see the website?
Let me see.
There.
Yeah, it popped up
Mm.
on our end.
Do you see it?
I don't know. I'm not savvy enough to figure out. I can barely see the
It's in the Zoom chat. It came
Yeah,
through.
so Zoom chat.
Oh, in the Zoom chat. Okay, I will. Once we get off, I'll post it in the comments of
Okay.
the live stream.
Okay. And I think now it's sort of there's before it was like qualitative, but like I say, we've upgraded, and so it was I was doing um well, it's just another sort of adaptation as you go, as you're trying to sort of find a solution. So when I first started doing this test, what I was doing was I would basically send people a PDF of any inclusions and a video of me just moving and finding the inclusions in their sample. And what I found was I would just get email after email as they would watch a bit more of the video that they make. And so what ended up happening is it turns into a sort of telehealth type thing. And what I do is I make a appointment and time zones allowing, and then it's usually an hour, 2 hours to go through their sample, field of view by field of view, and give the assessment. And now we can give, like I say, quantifiable counts and measures with the equipment upgrades we have. So that will, sorry to say, just include a price increase of what I was doing. If you just want a basic, is it there or not? I probably can do the current price, but if you want quantification, it's a bunch more steps that needs to be done. So I need to cover that somehow.
Yep. Yep. Well, it's impressive what you've been doing, and I appreciate you being the tip of the spear on this. And it's very sad that the government has abandoned us. But I'm very grateful to you. And Lindsay, we will get you to Japan, and very eager to see how this turns out. And
I'd like to return the compliment to you as well, Mary. I mean, like you were one of the first sort of lightning rod at the beginning of COVID for just watching the how sinister it all was with respect to how they were treating scientists and doctors who were speaking out. And I think your story is one of those people should reflect on. We don't want to go back to that if we ever can.
I agree. And both of you guys are both my heroes because without either one of you, I wouldn't be here. And I just want to say to my injured, just keep fighting. It's been a long and hell-bound road, but I feel like we have a little bit of hope and light at the tunnel. And just thank you guys. And this is our Hail Mary. I want
Yeah.
my blood clean. I'll be
Yeah,
knocking on RFK's door every day when my blood is clean. I will not go away from the White House.
good, good. And like I say, in terms of metrics that we're seeing right now, it doesn't get better than this. And yeah, just let's fix Lindsay, and that's our test. But I'm confident now after our current run. I'm confident we can certainly improve our quality of life.
Wonderful. Well, I hope everybody will pitch in even just $5. They're literally 6,600 or people watching. If you donate $5, we can get Lindsay to Japan because it's not cheap, but it's also the most effective thing that we have thus far. So it gives me
And
great
it's just way less than the US, Mary. That's the thing.
Right. No, and I'm not saying I don't think it doesn't sound unreasonable at all. But it's still, but
yeah, there's still money involved, unfortunately.
right.
I wish I could just throw open the doors, but no, there's a full hospital that has to back up this treatment, right? And there's a whole ER department, and there's the doctor has to go there to give you a consult, and if they need MRIs or anything, that all that needs to be covered, right? And so, but
Hey, I'm not a rocket scientist, but I think they just uncovered a lot of fraud with the Somalis in Minnesota. Maybe we can borrow that money and put it to
Yeah.
good use. I don't know.
Yeah, when you think about it, just that in and of itself, right? The millions, billions, I hear.
Yeah.
Crazy.
Horrible.
Mhm.
Yeah, it's also, it pales in comparison to the fraud that Pfizer and Moderna have committed. So I
Mm.
just want to remind everybody that, yes, this is a big uncovering of fraud, but Pfizer's fraud is a lot worse.
Yeah, they should be paying for
Right,
Lindsay's treatment. That's the
right,
thing.
right, right.
But it will take years, and probably if we wait for them to pay, Lindsay unfortunately would be long gone. And
Yeah, we're not waiting on
let's
them.
no, no, we can fix Lindsay now, right? I'm confident of that. And with each patient we do, the confidence grows, and every marker we have just points in the right direction. Not just points. I mean, it's just landing right in the spot where you want it, which is, I don't want to say complete remission of those aggregations, but man, we're reducing them a lot.
One last question. Assuming this goes well, as we start to do a lot more patients, how hard would it be to get this procedure to the US?
Well, that's on the FDA, isn't it?
But
And
you said you're not giving a drug, right?
but they're still responsible for licensing of therapies, and so there is a treatment component in the column setup that is um, you could argue as pharmacological in action because of the binding agents that in there that make it disease specific. And like I say, in this one, we're using for polyneuropathy as the primary marker. So target the blood flow from the brain through the jugular and use polyneuropathy canisters as the secondary filter. And so far it's working great. So, but they have to approve that. They have to approve those filters. And
Well,
so
but we could run trials, right?
yeah, that's the thing, right? It should be like a protocol needs to be put together with this data and some exploratory grant, and so I don't know whether like an RO1 is really the right category because that's more sort of scientifically orientated, but um, some multi-center clinical trial needs to be put forward. And right now, literally hot off the press, we have the data. And I'm not hiding there's no secret source here. These are treatments that were well established. It's just how we're combining them that's unique in this instance. But yeah, I'm happy to um, contribute to that protocol write up and provide the data in this instance. And um, yeah, in a few weeks we'll have a whole bunch more.
Wonderful. Yeah. Well, keep us posted, and
Mm.
we'll have you back soon and see how the progress is going. Thank y'all so much. Lindsay, you have anything else to add?
No, I'm good. Just thank you for having us on. And you really are our Hail Mary, and we love you.
Yeah. Thank you, Mary.
Yeah. Well, great to meet you, and we'll see you soon, and good luck. We will get you to Japan. Don't worry.
Okay. Hope so.
Can't wait.
All right. Did that stop?
Not yet. I gotta hold on.